The fishing industry is worth almost £1 billion to the UK economy, with shellfish becoming an increasingly important part of that. Almost 25% of the current catch comes from shellfish, so crabs, lobsters, oysters and scallops. The work of my laboratory focuses on diseases and pathogens of those shellfish and really how they impact upon the catchability and value of that stock. Recent survey work has shown that the pathogens of those juvenile life stages can be quite different to those that we see in the adult population. And this paper really focuses on an important pathogen we've recently discovered infecting juvenile edible crabs, canceri, that feed into this fishery. So here we are in the English Channel. The English Channel it supports a very important fishery for the brown crab, or the edible crab, canceri. The fishery, the adults in the fishery just out here, they release larvae. The larvae come inshore and they settle on rocks that you can see behind us here, where they grow to juvenile life stages, probably 20 to 30 millimeters in carapace. When those animals get older, they move again offshore into the fishery, where they can potentially be recaptured, probably in 4 to 5 years' time. Survey work that we've been carrying out on this shoreline in the English Channel has shown that some of these juvenile life stages have very different pathogen profiles to the adults in the fishery. In particular, in this paper, we describe a parasite we name as Paramikrocytos canceri, which is very prevalent in the juveniles of this shoreline and absolutely absent from those offshore.
So here we have our juveniles of the edible crab canceri, just collected from the shoreline here in the English Channel. The parasite Paramikrocytos canceri specifically infects the antennal glands, or the bladder, of this young crab, and in about 60% of the animals on this shoreline. The parasite lives within the epithelial cells of the bladder, and specifically within those cells, and living next door to mitochondria, and presumably gaining energy from the cell itself. The life cycle of the parasite progresses from small uninucleate cells to large plasmodia, and those plasmodia contain many nuclei. And both of those parasite stages, the uninucleate stages and the plasmodia, can end up within the urine, presumably for release with the urine into the water. And some of the work in our laboratory has shown that actually incubation of these young crabs in water does lead to actually release of those parasite life stages. The pathology can be very severe, but currently we do not actually know the fate of these infected juvenile crabs and whether they make it into the fishery at all.
I'm David Bass, and this is Hannah Hartikainen in the Natural History Museum sequencing facility. We extracted DNA from the infected edible crab tissue and tried to amplify 18S genes from it using a range of group-specific parasite primers. Surprisingly, none of those gave products, so we carried out metagenomic sequencing instead on the mice here. We found two eukaryotic small subunit genes, one from the crab and one from a highly divergent eukaryote, which turned out to be sister to the oyster parasite Mikrocytos mackini. We call this Paramikrocytos canceri. We mined out other genes for phylogenetic analyses using sequence signatures to avoid long branch artifacts, and showed that the Mikrocytid clade is sister to Haplosporidians.
Using Mikrocytids group-specific 18S primers based on existing Mikrocytos sequences and our new Paramikrocytos sequence, we investigate the host range, distribution and diversity of this clade in DNA from environmental samples and common shoreline invertebrates. We show that P. canceri here is also closely associated with mussels, top shells, prawns, annelid worms and other crabs. We found more novel lineages in environmental DNA, including a clade from freshwater and brackish samples. These sequences are very rare or absent in sequence databases because they are so divergent. We also report a mackini-like infection for the first time in UK oysters. However, mackini-specific assays did not recognize this parasite, so we used our group-specific primers to show that it is a novel divergent Mikrocytos species and describe it as Mikrocytos mimicus. Environmental DNA is a powerful approach for understanding microbial diversity, distribution, and parasitic relationships, particularly using highly specific probes. However, we emphasize the importance of complementary methods to confirm parasitism: in situ hybridization, histopathology, and transmission electron microscopy.
Mikrocytids are understudied because they're so divergent, possibly the most genetically divergent eukaryotes known. They're tiny and indistinctive, so difficult to detect in standard screens. We analysed only a limited selection of marine invertebrates, so we now want to know how much more mikrocytid diversity there is, what their other hosts are, and what the environmental lineages are doing in both marine and freshwater ecosystems.